Today we are going to talk about a very interesting question, that why are neutrophils first and the most abundant leukocyte to arrive at the acute injury site, right? For example, if there is acute injury to this finger, right, and it might be a trauma or it might be infiltration of pathogenic organism, within few hours, right, you will find that lot of neutrophils have accumulated at the focus of injury, right? And these neutrophils are trying to phagocytose the offending organism or destroy or eliminate the offending organism, and also trying to remove the necrotic cells. Now, our question is that especially during the first 24 hours of the injury, why neutrophils dominate at the site of injury? Why not macrophages? Why not lymphocytes? Why not eosinophils? Why not basophils? Why the neutrophils, first of all, appear at the site of acute injury, and especially for first 24 hours, they remain the dominant cells. Macrophage start appearing after 24 hours, right? And as time passes by, then macrophages start accumulating at the site of injury. But let's see, first talk about the neutrophils. There are three important reasons why neutrophils are the first responder to acute injury and in such a big number, right? As you know that neutrophils are mainly produced and stored in bone marrow house. Neutrophils are basically produced and stored, right, in the bone marrow house, and from there they come to circulatory system, and they are normally present in our circulation. And in our circulation, neutrophils are the most abundant white cells. Neutrophils are most abundant white cells. So about 50 to 70% of leukocytes in circulation are neutrophils. Usually we show them that their nucleus is multilobated. These different lobes of the nucleus are connected by the chromatin. Now, neutrophils, the number one reason why neutrophils reach to the acutely injured tissue first of all, and such a big number one reason is that they are the most abundant white cells present in our circulatory system, right? Number one is most abundant white cells, leukocyte, WBCs, in circulatory system. Number two, out of the white cells, out of the leukocytes, neutrophils are most mobile. Their ability to come out of circulatory system, to extravasate from the circulatory system, and through their amoeboid movement rapidly go to the tissue, that point of the tissue where there is focus of injury. This ability to move is best developed in the neutrophils. Then less than that is present in macrophages, right? So second reason why neutrophils are the first responders in acute injury for the tissue is that they are the most, mobile cells, mobile white blood cells or leukocytes. So naturally, which white cells are present? For example, if this is the focus of injury in the tissue, it is very natural which white blood cells are most abundant, they will be coming in bigger number. Secondly, which are the most mobile, they will be also coming in the early stage in bigger number and more rapidly. And then the third most important cause. Not only they are most abundant, neutrophils are not only most mobile, the most important point which I want to highlight is that when tissue is injured, injured tissue, special type of chemical mediators are produced in high concentration. In injured tissue, when tissue is injured, from the injured cell membrane, from the mast cell, from the resident macrophages, from the endothelial cells, from all these cells, and even the bacteria which are present, from all these injured cells and bacteria, lot of chemical mediators are produced. As we know that mast cells, which are present in connective tissue of all cells, and they are specially well concentrated under the mucocutaneous membranes in the connective tissue and especially perivascularly. Now mast cells, whenever you injure a tissue, what will happen? Mast cells are very sensitive. They are very, very sensitive. Whenever they get perturbed or they get disturbed, their membranes release arachidonic acid, and from there, arachidonic acid derivatives like prostaglandins and leukotrienes will be produced. Mast cell will release the histamines. Bacteria itself will produce chemical mediators which can attract the neutrophils. Then resident macrophages will release different chemical mediators. So what happens? Whenever a tissue is injured, right? In injured tissues, locally, there is production of chemical mediators, right? And these different chemical mediators are going to attract the bring the changes in the blood vessels, right? Inflammatory changes in the blood vessels, which we say those changes are involved in inflammatory process. For example, in injured tissue and around that, blood vessels will dilate to bring the more blood in that tissue so that more white blood cells and more complements and more immunoglobulins and other defense mechanisms can come to the site. Then these chemical mediators will help the endothelial cells to slightly swell and make interendothelial gap through which protein-rich fluid will extravasate and exudation will occur. Then these chemical mediators will act on the leukocytes and help the leukocyte to stick or to become adherent to the wall of the injured tissue, blood vessels, especially endothelium of venules, and help the white blood cells to immigrate out of vascular system and move chemotaxis, move towards the focus of injury. Now all these vascular events of inflammation and cellular events of inflammation, which we will discuss in detail later, all these events are controlled by chemical mediators. Right? Now, at the site of acute injury, those chemical mediators which are specially chemoattractant for neutrophils and chemoactivator for the neutrophil are produced first of all. Again, I will repeat it, it's worth repeating. At the site of acute injury, right? For example, if here is the acute injury, at the site of acute injury, the chemical mediators which act as chemoattractant to the neutrophils and also act as chemoactivator of the neutrophils, such chemical mediators are produced very rapidly and very big concentration. That is third most important cause, that neutrophils are attracted at very early stage and very strongly towards the focus of injury, right? So I would say the third cause is that chemical mediators, right, which act as chemoattractant and chemoactivator of neutrophils. For the neutrophils are produced first of all at the site of acute injury and in very high concentration. That is why neutrophils are very happy to reach there first of all, and they become first responder, because the first call of injury, call from the injured cell or injured tissue, is coming to the neutrophils, right? For example, these are some of the very important chemoattractant and chemoactivators of neutrophils. These are the chemical mediators. This is the list of chemical mediators which specially love to recruit the neutrophil out of the vascular system at the site of injury towards the focus of injury. Now these chemical mediators, like they are special. For example, leukotriene B4, which is produced by the cell membranes of the mast cells, phospholipids of cell membranes of the mast cells, cell membranes of the macrophages, cell membranes of the neutrophils itself, and even cell membranes of the endothelial cells, right? At the site of injury, the injured mast cells, injured resident macrophages which are previously there, and injured endothelial cells, and even newly arrived neutrophil, all of their membranes of such activated and injured cells, a special enzyme is activated. All of you know the name of that enzyme, phospholipase A2. For example, here is a mast cell. Here is a mast cell, and what will happen, that this is mast cell lipid bilayer. Within it, there are phospholipid, especially diacylglycerol, right? What happens when this membrane is injured, mast cell membrane, or neutrophil membrane, or macrophages membrane, or endothelial membrane, right? What will happen, within the membrane there is an enzyme, and this enzyme is called phospholipase A2. This phospholipase A2 will attack its own membrane. Phospholipase A2 will attack its own membrane, especially diacylglycerol molecule, which are part of the membrane phospholipids. And when phospholipids, diacylglycerol, that is damaged by the phospholipase A2 or acted upon by the phospholipase A2, then phospholipids of the membrane will release arachidonic acid, right? And as you know, arachidonic acid can go through cyclooxygenase pathway and lipooxygenase pathway. This cyclooxygenase pathway will end up into prostaglandins, prostacyclins, and thromboxane A2. And lipooxygenase pathway, this was cyclooxygenase pathway, lipooxygenase pathway will end up into leukotrienes, right? I will not go into detail of this pathway. We will have a separate lecture on that. But what I'm trying to tell you, that whenever tissue is acutely injured, at the site of injury, mast cells, resident macrophages, newly arriving neutrophils, local endothelial cell, all cells, their membranes are activated after injury, and these membranes are activated by other cytokines. These membranes are activated by the bacterial products. These membranes are activated by the physical injury. So any type of injury, when we say membrane is activated, what we really mean? That phospholipase A2 in the membrane is activated. And what it will do? It will act on the membrane phospholipids, its own phospholipids, especially diacylglycerol, and break it down into arachidonic acid. And arachidonic acid can go through two metabolic pathways, cyclooxygenase pathway, which end up into prostaglandins. Some prostaglandins act as vasodilator, right? So that local blood vessels dilate, arterioles dilate, and bring more blood in the acutely injured tissue. Some prostacyclins, they are acting as, I will not go into detail of prostacyclins and thromboxane A2, but it's worth mentioning that prostacyclin, which is prostaglandin I2, disperse the platelets, and thromboxane A2 aggregate the platelet. And prostacyclins is vasodilator and thromboxane A2 is vasoconstrictor. But right now we will not discuss in detail about that. I will just say that out of arachidonic acid, cyclooxygenase pathway will produce prostaglandins, and that will lead to vasodilation or arteriolodilation. But more important for the leukocyte, that when arachidonic acid goes under lipooxygenase pathway, leukotrienes are produced, leukotriene A is produced, then some of the leukotriene A will convert into leukotriene B, and some of the other leukotriene A will convert into leukotriene C, leukotriene D, and leukotriene C4, D4, and E4. Now, out of these leukotrienes, for neutrophil, leukotriene B4 is very important. So, what we can say, I'm just taking one example of chemoattractant for the neutrophil, leukotriene B4. That tissue injured, mast cell membrane, endothelial membranes, resident macrophages membranes, and incoming neutrophil membranes, they start releasing leukotriene B4 along with many other arachidonic acid derivatives. These leukotriene B4, you know what they do? Leukotriene B4 will go to they will control the multiple steps of recruitment of neutrophils, especially. Leukotriene 4 produces the trains of the neutrophils coming out of the blood vessels. For example, leukotriene B4, these are endothelial cells, right? And let's suppose here is neutrophil. They will, under the influence of leukotriene B4, endothelial cells will start expressing selectin molecules, and then integrin molecules, E-selectins and P-selectins are expressed on the endothelial cells, and integrin molecules which are present on the neutrophils, integrin molecules convert into high affinity state. So first, selectin dependent interaction occur between the endothelial cells and neutrophils start developing loose attachment with the local endothelium. Then integrin dependent activity occur, and neutrophils strongly attach with the, or become adherent with the, or stable adherence with the endothelial lining, right? And then After that, neutrophils will extravasate. They will immigrate out. They will come out of this. And then they will move towards the focus of injury. The directional movement of neutrophils towards the chemoattractants. This direction movement, which is moving after the extravasation from the circulatory system, neutrophils start moving towards the focus of injury, determined by the progressively increasing concentration of the chemoattractant like leukotriene B4 or platelet activating factor or C5a or interleukin 8 and others. This directional movement is called chemotaxis. What is chemotaxis? Directional locomotion of the cells, right? Directional locomotion of the cells determined by the concentration of a chemical mediator. Now, for example, if we take the steps of neutrophil arriving from the vascular system to the site of injury outside the vessel, we say neutrophil will first of all make loose adherence to the endothelium, which is selectin dependent, then tight adherence to the endothelium, that is integrin related interactions, then go out of what is this interendothelial gaps or through the endothelial lining, they extravasate, that is dependent on PCAM. First it is selectin dependent. Loose attachment is selectin dependent. Then tight adherence or adhesion is integrin dependent. And then they move out, that is PCAM dependent. Now, expression of selectins, enhancing the activity of integrins and extravasation through PCAM, and after that chemotaxis, right? And once the chemotaxis, under the influence of chemotactic agent, when neutrophils reach at the site of injury where they find the offending agent, they are ready to fight. They are super activated there. Then they can fight either by phagocytosis of microbe. Let's suppose there is bacteria. Either they will phagocytose the bacteria or they will have lysosomal activity and kill the bacteria, or oxygen burst and reactive oxygen species are there, or later on I will explain that a new mechanism has been discovered about neutrophil action, that neutrophil makes nets. Nets stand for neutrophil extracellular traps. You know what neutrophils do? If I'm a neutrophil, neutrophils are not going to live long in the battlefield, right? Usually they survive in the blood only 1 day to some authorities say even less than 1 day to you can say 5 hours to 135 hours while they are circulating. But once they come out and they are fighting with the microbes or with the offending agent, then their lifespan is very much reduced. So anyway, while they are fighting and trying to control the offending agent, they are going to die. They are acting as suicide bombers. They are going to die themselves and trying to kill the offending agents. Now, new mechanism has been discovered. Not only neutrophil can phagocytose the offending agent and kill the offending agent by lysosomal action or by producing reactive oxygen species. Another mechanism is out of the neutrophil, special molecular ropes come out, molecular chains come out, which are made of chromatin, and they are plastered with destructive hydrolytic enzymes. And these molecular chains which come out of, or fibrils which come out of neutrophil, they grab the offending agent, right? and trap them. And such traps are called neutrophil extracellular traps. Usually these traps are used for those offending agents which neutrophil is unable to phagocytose. So it can just produce chromatin extension out of the cell, and these extensions or fibrils are plastered with or painted with very, you can say, with poison, with very destructive enzymes, and they will make a network around the offending agent, and now offending agent cannot escape away. And this is in a cage, this is in a trap, and there they will keep on concentrating their destructive enzymes and reactive oxygen species and kill the organism there, offending organism. Now, so we can say that if neutrophils are, listen carefully, if neutrophils are first to reach at the site of injury, then they should be the first to make selectin mediated attachments, loose attachment. They should be the first to make integrin mediated tight attachment to the endothelial lining. They should be the first to extravasate. They should be first to undergo rapid chemotaxis process, and eventually they should be the first to reach at the site of injury and start their killer action by either phagocytosis or by phagolysosome fusion and digestion of the phagocytosed material, or by activation of special oxygen burst reactive oxygen species. And if they cannot do anything, they at least can extend the special ropes and chains out of their cell membrane. And these long fibrils made of chromatin painted with the poison of hydrolytic enzymes will trap the offending agent, which is called neutrophil extracellular traps. Now, these cellular events, right, they are all controlled by the chemical mediators. These cellular events, all of them are controlled by chemical mediators. And the chemical mediators which produce such cellular event for the neutrophil are first to be produced at the site of injury, and they are in higher concentration. That is why neutrophils first of all arrive at the site of injury and in a very big number, right? Now, what are the chemoattractants? What are the chemical mediators, especially for the neutrophils? Chemoattraction and chemostimulation. They are leukotrienes B4, which are produced by the membranes, right, as I told you, from certain cells, or also there is release of platelet activating factor. Listen very carefully, that platelet activating factor, even though it was originally discovered in platelets, but actually platelet activating factor is also derived from membrane lipid and also from the mast cells, neutrophils, macrophages membrane, endothelial membrane, and of course platelets. Now listen, leukotriene B4 is also derived from the membrane, platelet activating factor is also derived from the membrane. The difference, common thing in them is that both of them are derived from the phospholipids of membranes of mast cells. Both of them are also derived from membranes of the neutrophils. They are also derived from the membranes of resident macrophages. They are also derived, both of them are also derived from the membranes of endothelial cells. But platelet activating factor is, in addition to other cells, also derived from platelets. So both of them are lipid derivative, membrane lipid derivative. The difference is that this is derived from arachidonic acid pathway, from lipo-oxygenase pathway, leukotriene B4, and this is by other pathway. Now, what is functional difference in them? The functional difference is that leukotriene B4 mainly act on the cellular events in acute inflammation, especially for neutrophils. What are the cellular events? As I told you, there is rolling, loose attachment by selectins, right? This is activated by leukotriene B4, also by the platelet activating factor. Next cellular event is integrin mediated tight attachment. That is also promoted by leukotriene B4 and platelet activating factor. Then extravasation and chemotaxis of neutrophils, that is also activated by leukotriene B4 and platelet activating factor. Then once neutrophil reach at the site of injury, as I told you previously, they become super active for killing action, right? The way where there is phagocytosis, lysosome mediated killing, or respiratory burst which produces reactive oxygen species, or nets are produced. All these things are also promoted by leukotrienes B4 and platelet activating factor. So what I can say, I would say it an easy word to remember, that leukotriene B4 and platelet activating factor, both of them accelerate or promote the all cellular events related with the neutrophil at the site of acute injury. Both of them are involved in neutrophil rolling, selectin dependent, neutrophil stable attachment or tight attachment, integrin dependent, extravasation, PCAM dependent, and chemotaxis, and eventually full active killing action, right? But there is a difference. Platelet activating factor has not only promoting the cellular events, but in addition to that, it also promotes the vascular events at the site of acute inflammation. The vascular events are what? Vasodilation and interendothelial gaps and increased permeability and exudation. That due to increased permeability, protein-rich fluid will leak out from circulatory system into the injured site, right? Which will bring more antibodies and which will bring more complements and other important molecules to deal with the offending agent, right? So what I'm saying, now let me make a little more clarity. Leukotriene B4, we learned well that it can activate cellular events related with the neutrophil very strongly. Or we can say one event, we just talk about the chemotaxis. Leukotriene B4 is a very strong chemotactic agent for neutrophil. But it is also chemotactic agent for macrophages. But not strong, moderately. It is also chemotactic agent for eosinophil, but very weak. So it means the chemical mediator like leukotriene B4, which is very rapidly produced during the acute inflammation, at the site of the acute inflammation, has strong chemoattraction for neutrophil, moderate chemoattraction for monocyte macrophage system, and weak chemoattraction for weak recruitment stimulus for eosinophil. So this is what we have made up to now, that why when there is acute injury, neutrophils are the first white blood cells which act as first responders to reach at the site of injury, not only most rapidly but also in big number. The answer is number one answer. Why? Because neutrophils are most abundant white blood cells in circulation. So they must come out first of all in bigger number. Secondly, they are most mobile, because for mobility these cells have to show amoeboid movement. They should be able to make rapidly pseudopods. And cytoskeleton of neutrophil is far more agile and rapidly working as compared to other white cells. So neutrophil reached to the site of injury also due to their agility, due to their ability to move fast. And then last but not least, very important, only good doctor answered this. The neutrophils reach first of all at the site of injury because at the site of acute injury, the chemical mediators which recruit the neutrophils and activate the neutrophils are produced first of all and in bigger concentration. Right? And these chemical mediators, two of them I told you in detail, like leukotriene B4 and also platelet activating factor. Then another very important chemical mediator is complement C5a. Now, normally complement factors are produced in the liver and they are secreted into circulatory system. And the site of acute injury, complement factors leak out due to increased permeability at injured site. Their complement, which are plasma derived, liver made, but at the site of injury they are coming from plasma. These complement factors are activated by different mechanisms which I will not discuss here. But one of the complement factor number 5, during activation cascade, it breaks down C5a and C5b. Right? C5a is also a very good chemotactic agent for neutrophils. Rather, I must say that C5a also lead to activation of endothelium, right? C5a also lead to activation of endothelium. When endothelial cells contract, interendothelial gap appear and more fluid protein rich exudate come out. Secondly, C5a is also very strong chemotactic agent, and C5a also activate the killer activity of the neutrophils. So what we have seen, these are the chemical mediators which are produced at the site of the injury to recruit the neutrophils and activate the neutrophils. These two are coming from membranes of mast cells, endothelial cells, resident macrophages, and from the neutrophils themselves. This is plasma derived C5a. Then there is interleukin 8. Even though I have mentioned it at position number 4, but in your gray matter, interleukin 8 should be number 1 when we talk about neutrophil chemotaxis. Neutrophil chemotaxis, yes, interleukin 8. This is so powerful chemotactic agent for the neutrophil that it is called chemokines. It's small molecular soluble protein, and this is called chemokine because it's a chemical substance which increases kinetics of the white blood cells. And interleukin 8 is specially active on the neutrophils, right? But the question is that from where the interleukin 8 is being produced, right? From where the interleukin 8 is being produced. One of the very rich source of interleukin 8 is macrophages. Macrophages which are present at the site of injury, right? Macrophages can produce what? Interleukin 8. You remember these two are being produced by the mast cells. Interleukin 8 can also be produced by the mast cells. Leukotriene B4 and platelet activating was also produced from the membranes of, yes, endothelial cells. Interleukin 8 can also be produced by the endothelial cells. Then leukotriene B4 and platelet activating factor was produced by macrophages. Interleukin 8 can also be produced by the macrophages, right? So all three of them can be produced by the mast cells and also from the endothelial cells, also from the macrophages. Interleukin 8 has one thing special. It can be specially produced by the fibrocytes and fibroblasts. So it means that not only it is produced during acute inflammation, but when inflammation becomes prolonged, still it is produced by activated fibroblast. Now you look at it. What is happening? If someone asks, tell us one single most important, most powerful neutrophil chemoattractant, just tell interleukin 8. Right? Interleukin 8. It is a type of a cytokine, right, which is produced by mast cells, macrophages, endothelial cells, and fibroblasts. Right? And it is so powerful in attracting the neutrophil that it is also called neutrophil chemotactic factor. Interleukin 8 is also called neutrophil chemotactic factor, right? And these cells can also produce, I told you mast cell, which cells produce this? Mast cells, I'm repeating it, macrophages, yes, endothelial cells and fibroblasts. You know, interleukin 8 is also produced by a very special stimulus, that is by the pathogens. We call it PAMPs. Pathogen associated molecular patterns, pathogen associated molecular pattern, right? Especially gram-negative bacteria, they produce some molecules, right? And like lipopolysaccharides, right? Or flagellin from flagella, right? So these are components of pathogens, pathogen-associated molecular patterns. These patterns act on the target cells. These molecules, when they bind with the target cell have special receptors. Target cell mean mast cells, endothelial cells, macrophages, fibroblasts. They have special receptors for such pathogen-associated molecular pattern, special molecules. Those receptors are called pattern recognition receptors. For example, toll-like receptors. For example, I'm a macrophage, you are a pathogen. Sorry, but you are a pathogen. And I'm the macrophage. If some molecule touch me, those molecules are commonly present in so many pathogens. They are shared by many pathogens. For example, lipopolysaccharides or endotoxins are shared by almost all gram-negative bacteria, right? Or teichoic acid is shared by gram-positive bacteria. Or flagellin is present in many flagellated bacteria. So if such product from the pathogen touch the macrophage at this point, now these are commonly present, these flagellin or lipopolysaccharide endotoxins or teichoic acid are commonly present in many pathogens. So these molecules are pathogen-associated molecular patterns. On the macrophage, they will bind with a special group of receptors. That group of receptor is called pattern recognition receptor, because these receptors recognize those molecules which have some fingerprints that there is some pathogen is around. So they recognize the pattern of molecule which are present in pathogen. So we call them pathogen recognizing receptor. These receptors have many subclasses. One of them is very famous, toll-like receptor, and there is another NLR, node-like receptor, and many other. When these receptors are activated on the macrophages, they will give signal inside the macrophage, activate transcriptional factor like NF-kappa B, and those transcriptional factor will go into macrophage DNA and activate the genes which are related to produce cytokines, and one of those genes is gene for interleukin 8. And once interleukin 8 is secreted, synthesized and released by macrophages or endothelial cells, right, or mast cells or fibroblast, right? So it means these cells which are producing interleukin 8, basically they recognize the many signals. Very important signal among that was signals from the molecular signals coming from the pathogenic bacteria and acting on the pattern recognition receptors which give signals inside the cell and activate the genes related with cytokine production, especially interleukin 8. And interleukin 8 is a very powerful stimulator for all the cellular events related with the recruitment and activation of neutrophils. What are those events? You already know. What was that? Loose attachment, selectin dependent. This is promoted by interleukin 8. Even endothelial cell have preformed interleukin 8 and they release that when they are injured. Preformed, stored in special you can say vacuoles called Weibel-Pollard bodies. Weibel, okay, I will write it here. Bodies. Within the endothelial cell, there are Weibel-Pollard bodies, and when endothelial cells are activated or injured, they release content of Weibel-Pollard bodies, and these bodies not only release some type of selectins, but they also release interleukin 8. So that as soon as possible, neutrophil related activity can be started in the acutely injured tissue, right? So what we have learned that interleukin 8 is one of the most powerful, rather most powerful chemotactic agent for what? For neutrophils, right? Then we come to another factor, and this factor is tumor necrotic factor. Tumor necrotic factor. Tumor necrotic factor can be produced by many cells. Now this is very important. This is a cytokine and it can be produced by many cells. First of all, remember your initial cells which were producing leukotriene B4, platelet acting factor and interleukin 8. The mast cells, macrophages, endothelial cells, all these cells can produce tumor necrotic factor. Plus, tumor necrotic factor can also be produced by T lymphocytes and natural killer cells and fibroblast and endothelial cell. Now let me tell you, where there is acute injury, mast cell, resident macrophages, endothelial cells, right? They can produce leukotriene B4, platelet activating factor, interleukin 8, even tumor necrotic factor, right? And tumor necrotic factor can be additionally produced by endothelial cells and T cells and natural killer cells. And when these cells produce tumor necrotic factor, answer is when these cells are exposed to PAMPs or exposed to DAMPs. These are, I have already told you PAMPs. PAMPs are pattern associated, what is this? Molecular, pathogen associated molecular patterns. Pathogen associated. It means if in my finger there is some pathogen like some pyogenic bacteria present, if they are releasing some molecule, right? And for example, a gram-negative bacteria is there and they are producing endotoxins or releasing endotoxins or lipopolysaccharides, or there's staphylococci releasing teichoic acid. They are examples of PAMPs. DAMPs are damage associated molecular patterns. What damage? Our local tissue damage. For example, when our local tissue is damaged, local cells are damaged, cell membranes are disrupted, mitochondria will be free, and those mitochondria will be disrupted, and mitochondrial DNA act as indicator of damage to the local tissue. Mitochondrial DNA act as fingerprint of or indicator of or signal of local tissue damage. So mitochondrial DNA, injured mitochondrial DNA at the site of acute injury is acting as damage associated molecular patterns. So what happens? Now let me tell you how tumor necrotic factor is produced. Imagine I'm a cell who has capability to produce tumor necrotic factor. So what kind of cell I can be? I can be mast cell. I can be macrophage, right? I can be endothelial cell. I can be fibroblast, right? I can be natural killer cell, or I can be T lymphocyte. So these cells are capable of producing tumor necrotic factor, which is a cytokine, very important cytokine. But how these cells know when to produce tumor necrotic factor? Answer is that they are having pattern recognition receptors. I have pattern recognition receptors. And these pattern recognition receptors can recognize PAMPs and also can recognize DAMPs. And whenever in these cells, if I'm a macrophage and I'm receiving PAMPs and DAMPs, evidence of pathogens presence and evidence of my own tissue injury. When I'm receiving PAMPs and DAMPs, my pattern recognizing receptors, toll-like receptors, and their other associated receptors are activated. They will give intracellular signal. Those signals will simply shoot towards the nucleus of the macrophage, nucleus of the mast cell, nucleus of the endothelial cell, nucleus of the fibroblast, nucleus of natural killer cell, nucleus of T lymphocytes. So all these signals will go to these cells and they will activate NF-kappa B. NF-kappa B. This is a transcriptional factor. This is a special key. This will bind with those genes. This is the transcriptional factor which will activate the transcription of those genes which are related with the production of messenger RNA for tumor necrotic factor, interleukin 8 and many other cytokines like interleukin 1. So all these cells which are present in acutely injured tissue, right? But this tumor necrotic factor can be produced chronically also. But in acute situation also, when tumor necrotic factor is producing cells that are activated, how they are activated? They can be produced by the presence of PAMPs. They can be produced by the presence of DAMPs. Even they can be produced, yes, tumor necrotic factor cells can be forced to produce tumor necrotic factor and interleukin 1 when they are antigen antibody complexes, right? Immunological reactions. Or these cells can also produce tumor necrotic factor when they are exposed to many other cytokines. It means I'm macrophage. I'm exposed to the DAMPs or I'm exposed to the DAMPs. DAMPs are evidence of local cell injury. PAMPs, damage of presence, indicator of presence of pathogen around, or immunological immune complexes, or some other cytokines come to me, a macrophage. Which cytokines? Many cytokines like interleukin 1, interleukin 6, interleukin 18, gamma interferon, beta interferon, all of them when act on this macrophage start massively producing tumor necrotic factor. And this action not only happened to macrophage, I told you this can happen with the mast cell, with the endothelial cell, with the macrophages, with the dendritic cells. This doesn't happen with the neutrophils, right? And this happens with the fibroblasts. This can happen to T cells and natural killer cells. And all these cells producing tumor necrotic factor. And this tumor necrotic factor has many functions, right? One of the function is that tumor necrotic factor basically increase all the cellular events involved in recruitment and activation of neutrophils. It means tumor necrotic factor at the site of acute injury will produce more neutrophil adhesion with the endothelium. It means that more adhesion, more extravasation of neutrophils, more chemotaxis of neutrophil, more killing activity of neutrophils, right? So what we have seen that which cells produce tumor necrotic factor, what are the stimuli to produce the tumor necrotic factor, and once tumor necrotic factor is produced, what it is doing to recruit the neutrophil. Especially loves to recruit neutrophil during the acute phase, right? Then, after that, the last thing which I would say is formulated bacterial peptide. When bacteria are there, some of the bacterial peptide are formulated. These formulated bacterial peptide act as PAMPs. What is PAMPs? Pathogen associated molecular pattern. And you know, PAMPs can activate formulated bacterial peptide act as PAMPs, right? And these PAMPs can activate production of interleukin 8, tumor necrotic factor, and even they can directly act on the neutrophil and increase chemotaxis by the neutrophil. So, formulated bacterial peptide can increase the chemotaxis of neutrophils, number one, directly, number two, via acting on other cells and forcing the other cell to produce interleukin 8. So what we have learned up to now, a brief review. We were trying to answer why at the site of acute injury, usually neutrophils are first to reach there and in biggest number. Answer is number one, neutrophils are most abundant leukocyte present in our circulatory system. Number two, neutrophils are the most mobile leukocytes. Number three, the chemical mediators which act on for the recruitment of neutrophils and also not only recruitment of the neutrophil but also activation of neutrophils are produced first of all and in very high concentration at the site of injury, and these chemical mediators are leukotriene B4, platelet activating factor C5a, very powerful interleukin 8, tumor necrotic factor, and if bacteria are there, then might be there a formulated bacterial peptide. So this is the classical answer. Now we come to another question. What are the cells of acute inflammation? Classically, every doctor will say neutrophils are the cells of acute inflammation. This is right. This answer is right because actually neutrophils, as I told you, they are first to arrive there, but they have a short lifespan. Neutrophils have short lifespan. So within few hours and within 1-2 days, they undergo apoptosis. They are killed. And then other cytokines which are being produced, they start recruiting more hardy, more powerful phagocytic cells, which are macrophages. So as for first 24 hours of injury, these are the neutrophils which are the dominant cell at the site of acute injury. After 24 hours, then chemical mediator also start recruiting the monocytes from circulation, and when monocyte extravasate from circulation and come into tissue, we call them macrophages. So after 24 hours to 72 hours, there is a mix of neutrophil with macrophages at the site of acute injury. After that, if acute injury is not resolved, then gradually macrophages population start increasing. Why? There are two reasons for that. Even though macrophages are less in number in blood, even though they are late to arrive, even though macrophages are slow to arrive, but they have some important thing. You know what? Neutrophil reach fast, neutrophil reach in big number, but they die. They are suicide bombers. But macrophages are like tanks. They reach at the site of injury slowly, but they keep on coming. If injury persists, they keep on coming and accumulating it. So gradually they become dominant. Even macrophages in the tissue can multiply. Remember, mature neutrophil cannot proliferate, but mature macrophages can reopen their genes related with proliferation and they proliferate. So due to, as time passes by, at injury site, neutrophil number might become less and macrophages start dominating the picture, histopathological picture. Why? Because even though macrophages are slow to come, late to come initially in the less number, but they keep on accumulating because they don't die easily and they can proliferate locally. And then as time passes by, chemical mediators for neutrophil become less effective and less in concentration, and chemical mediators for attraction and activation of macrophages in the tissue, such chemical mediators become more. So in acute inflammation at very early stage, you will mainly see neutrophils, but as time passes by, it may become a mix of neutrophils and macrophages. So someone may say, yes, the earliest champion of acute inflammation cell is neutrophil. But after 48 hours or 72 hours, macrophage has the significant role as a cell of acute inflammation. Right? Then, these are the two white blood cells. If you want to give one answer, cell of acute inflammation, you say one white blood cell, most important neutrophil. But if you want to give two answers, you say initially neutrophil, and within 24 hours to 72 hours, somewhere between, lot of macrophages arrive. Now, there are some notable exceptions. For example, if there is acute injury, but this is due to parasite, or it is IgE mediated allergic reaction, type 1 hypersensitivity reaction. If tissue is being injured but type 1 hypersensitivity reaction or IgE mediated injury, or there is parasite induced injury, the first cell to come is eosinophils. This is a notable exception, that when there is acute injury by IgE mediated allergic reaction or by parasite rather than neutrophil, usually these are eosinophils which first of all come, because in such circumstances the chemical mediator which is more abundantly produced than other chemical mediator is eutexin, which produces chemotaxis of eosinophils. Then another notable exception, very important notable exception, if there is acute injury due to any viral activity. Usually neutrophils don't respond to viral infections. So when there is acute injury due to viral infection, in most cases of viral infections, the first cell to reach there and deal with it are lymphocytes. So it means classically cell of acute injury is leukocyte of acute injury is neutrophil, but within 1 to 3 days macrophage also play a role. But if injury is caused by specifically by the parasite or by the IgE mediated allergic reaction, these are the eosinophils who are coming first. And if acute injury is due to viral infection, then these are the lymphocytes which are coming first of all. Then if someone asks you, tell us more cells of acute inflammation, we can still tell. Because up to now, the cells of acute inflammation we discussed were basically leukocytes. But you must remember that any tissue which is injured, not only neutrophil macrophages come over there, but there are many other cells which are also active there. Don't forget mast cell. In acute injury, they were also injured and they were playing their role. So mast cells, right? Resident macrophages, endothelial cells. So mast cell, resident macrophages, endothelial cells, they can also play, they actually play a role in what? Acute inflammation. So we can say, when someone asks you, what are the cells of acute inflammation? You say, as far as leukocytes are concerned, the first to arrive are neutrophil, and after 24 to 48 hours, second to arrive are macrophages in most cases. But if we talk about cells of acute inflammation, other than the white blood cells, we can talk about mast cells, endothelial cells, right? So these two are very important example that mast cells and endothelial cells play a big role in acute inflammation. Right? Here we stop today. In coming lecture, I will go into detail of acute inflammation cellular events, because we have already recorded a lecture previously some years back. I have already recorded a lecture on acute inflammation vascular events. Right? Now, upcoming lectures will be acute inflammation cellular events. There I will discuss into detail role of endothelium of inflamed tissue, selectin mediated actions, integrin mediated actions, PCAM mediated actions, and also I will discuss mechanism of chemotaxis, and eventually I will discuss in detail not only what is the process of phagocytosis, but I will also discuss about how phagosome lysosome fuse and how the respiratory burst and reactive oxygen species are produced, and what are reactive oxygen species and how do they kill the bacteria, offending organism. And very importantly, I will love to discuss, I'm in love with this concept, neutrophil extracellular traps. We will discuss into detail these things in upcoming lectures. Thank you very much.